listen. There it goes. You're all in listen-only mode, and so if you have questions during the webinar, please go ahead and either put them in the chat, or actually, you could probably raise your hand. So whatever feels organic in the moment, please go right ahead. Our speaker today is Dr. Ray Najjar from Penn State University, and he's going to be talking to us about a project called the Chesapeake Carbon and Alkalinity Study, or CHALK. Ray, since you're controlling the slides, would you go to the next one for me, please?
Sure.
So for those of you who may not be familiar with MACAN, we are the Mid-Atlantic Coastal Acidification Network, and we are a virtual nexus of scientists, federal, state, and tribal agency representatives, resource managers, and industry partners who all kind of get together and coordinate to guide regional observations around acidification, both ocean and coastal. We all collaborate on pushing forward best practices, looking at different types of modeling and research methods that can be translated across the region. MACAN's goals are to better understand the drivers of coastal and ocean acidification. We hope to someday be able to very accurately predict the consequences of acidification on marine resources, and we're doing pretty well at that. We also, like I said, coordinate best practices for monitoring and data collection so that we are all communicating amongst each other in hopes of unified methodologies and results. And of course, one of our favorite things to do is outreach and education, and we try to include as many different types of stakeholders and ocean users as we can to help communicate solutions to acidification as well as mitigation strategies. So from here, I'm going to turn it over to Ray, and we're really excited to have you join us today. Thank you.
Well, thanks, Janet, and thanks everyone for joining us here today. I'm happy to be presenting a project that I've been leading for the past, I think we're coming up to 2 years now, called the Chesapeake Carbon and Alkalinity Study, or CHALK. And as you can see from this introduction slide, this is research that's funded by the National Science Foundation. Both the chemical and biological oceanography programs at NSF are supporting this. And yeah, it involves many investigators, as you can see here, from Penn State, which is where I am, here in central Pennsylvania, folks from the Virginia Institute of Marine Science, Chesapeake Biological Laboratory, Woods Hole Oceanographic Institution, St. Mary's College of Maryland, the United States Geological Survey, and well, I put North Carolina State University there because we had an REU student who participated in the project. That's research experiences for undergraduates student. So I'll be showing some of the work from that student as well, since it's related to the project. I'm going to give some introduction and motivation initially. Then I'll talk about broadly about our project. And then I'm basically going to go through almost like individual presentations from various components of the project. This is really the first time I'm presenting in like one presentation the results from multiple aspects of the project. So it's a bit of it's perhaps not as synthesized as I would like it to be. I hope you'll bear with me there. But I think we do have a lot of interesting results that I hope you'll be interested in. And I will stop periodically to see if there are any questions that show up in the chat. So feel free to look to use that. And Janet, I think I'll ask you to monitor the chat, because the way my screen is set up, I really can't see the chat. So is that okay?
Yeah, that's fine. Also, I just
Yeah.
wanted to point out, if you have questions, you need to put them in the Q&A. I don't know.
Q&A, not the chat, right? Yeah. Okay. Yeah, so I'll stop periodically and address questions if there are any that happen to come up. But yeah, do feel free to use that Q&A along the way, or hand raising feature if that's possible. So I'm going to start here with kind of big picture about the global carbon cycle, which is of course important for a number of things, including the regulation of climate. And this is a diagram from a recent synthesis paper that I contributed to, led by Pierre Renier. And it sort of describes a part of the carbon cycle that's often overlooked. And it's what we would call the land to ocean aquatic continuum. And, usually the carbon cycle is discussed in terms of like what's happening on land and what's happening in the ocean. And in this synthesis paper, we instead put our emphasis on, what connects those two reservoirs. That would be inland waters, like rivers and reservoirs, so forth. And that water makes its way to the coast, passes through estuaries. That water is influenced by tidal wetlands, and goes through continental shelf waters and ends out into the open ocean. And as you can see, there's a lot of carbon fluxes that occur along this route of water being carried to the open ocean. Some carbon is buried. There's a fair bit of outgassing, particularly in inland waters. There's some uptake of CO2, particularly by tidal wetlands. There's uptake by continental shelf waters, and so forth. I'll zoom in here on just what's happening near shore. And I sort of broke up some of these fluxes, which actually in the paper, we lumped estuaries and tidal wetlands together. Of course, they operate very differently. Estuaries actually outgas a fair bit of carbon, and tidal wetlands take up even more. And then there's quite a bit of burial of carbon in estuaries and in particularly tidal wetlands, as well as in continental shelf waters. So there are a lot of transformations or exchanges of carbon, I should say, between the water and the sediments, as well as in the atmosphere. And this is in contrast to earlier views of the way carbon went from land to sea. It was more viewed as like a pipe, with rivers just sort of dumping carbon out into the sort of, beyond shelf waters into the open ocean. All right, so that's a little bit of the carbon background. This talk is also about alkalinity. It's a little bit of a sticky concept, but really very important to aquatic carbon cycling and important to issues of ocean acidification. I'm sure it's a concept, an idea familiar to many of you, but I think it's still worth going over some of the basics. It's really the acid neutralizing capacity of a body of water, and that's how it's measured, by a titration with acid. And it does regulate pH. Also can be thought of as this way, as regulating the speciation of inorganic carbon, dissolved inorganic carbon, among the three main species. That's carbon dioxide, a bicarbonate ion, and carbonate ion. It should also be noted that it's a very commonly measured carbonate system variable in estuaries. And that's relevant for our project because we can mine historical measurements of alkalinity and pH in estuaries to learn something about long-term change of carbon cycling in estuaries. It's important for these other two reasons as well. Unlike pH or carbonate ion or carbon dioxide itself, it's conservative with respect to changes in temperature, salinity, and pressure. And also, unlike those three variables, it responds in a very straightforward manner to aquatic processes, particularly those involving calcium, nitrogen, and sulfur. And so there's kind of two definitions of alkalinity, and really the one that's probably of greater use for this presentation is this one here, which says if you're going to change the alkalinity of the water, you have to change the calcium ions, nitrogen, nitrate, particularly, or ammonia ions, or sulfate. So this links to very particular processes in aquatic systems. So here's an example. This is kind of a common a plot that you'll see. Alkalinity and dissolved inorganic carbon will be master variables. And from those two variables, you can compute a whole bunch of things, like what's shown here in the solid lines is the carbon dioxide concentration. And what you'll see is, as the dissolved inorganic carbon, that is the total sum of carbon dioxide, bicarbonate, carbonate ions, as that sum increases, the amount of carbon dioxide increases. It's fairly intuitive. On the other hand, as the alkalinity goes up, the CO2 actually goes down. It's not as easy to understand, and we don't have to go through it here. But it has an important effect on the carbon dioxide concentration, and therefore the partial pressure of CO2 in seawater. And also on this plot are shown particular processes in aquatic systems that are important. For example, air-sea gas exchange is shown as CO2 invasion. It's coming from the atmosphere, or CO2 release, being released to the atmosphere. And you can see when you add or remove CO2 from the water that way, the alkalinity is actually unchanged, but the dissolved inorganic carbon changes. Then we also have the processes of organic matter production and remineralization, basically photosynthesis and respiration, which mostly affect the dissolved organic carbon and have a small effect on alkalinity. And it's through nitrate that it has that impact. And then we see here some arrows that describe the impact of calcium carbonate dissolution and calcification. And you can see that, like, for example, calcium carbonate dissolution will increase both the dissolved inorganic carbon and the alkalinity, but the net effect will actually be to decrease the carbon dioxide concentration. So, and that's not completely obvious, without going through a diagram like this. Okay, and in addition to the roles I mentioned, also carbon and alkalinity are relevant for numerous important estuarine phenomena. We already talked a little bit about metabolism. They can be traces for understanding metabolism. Of course, they're important for understanding acidification, air-water CO2 exchange, which we discussed a little bit. They're relevant for hypoxia. Hypoxia is basically caused by remineralization of organic carbon. They can impact water clarity when in trying to understand marsh inundation and carbon sequestration. We often want to think in terms of carbon and alkalinity units. So one thing that we learned from an earlier study a couple of years ago is that the Chesapeake Bay, which is as you know, is an estuary, large estuary in the Mid-Atlantic region of the United States, has quite a large range of non-conservative alkalinity behavior. And it's best shown in this plot here on the left, which for several tributaries of the Chesapeake Bay, including the main stem of the bay, which is indicated by the Susquehanna River, and some of the larger tributaries like the Potomac, on the western shore, and the Rappahannock River, and so forth. You can see average alkalinity plotted versus salinity here. And what we expect for a conservative quantity in an estuary is that the quantity will plot along a straight line. And you can see, while there are some sort of straight lines in here, they're generally either bowing upward, as you see for the York River here, or kind of bowing downward, as you can see really clearly here for the Potomac River estuary. So what this tells you is that there's a sink of alkalinity in the Potomac River estuary, and there's a source of alkalinity, for example, in the York and the Mattaponi. The York, both the New York River estuary. And we also did a simple calculation of the fraction of the riverine alkalinity that was consumed in each of these estuaries. And they plot along this fairly a nice relationship here, which is as the river alkalinity goes up, you can see that the fraction that gets consumed in the estuary changes in a very systematic way. So rivers with low alkalinity have actually an alkalinity source. It's a negative consumption. And rivers with high alkalinity, like the Potomac, have an alkalinity sink. And you can see that the riverine end member here, which is shown in these little open circles, varies really quite dramatically among the different rivers. For the Potomac, the alkalinity is almost the same as seawater alkalinity. It's just a little bit below, maybe 20 or 30% below seawater alkalinity. Whereas, the York, the Mattaponi River is like an order of magnitude smaller than seawater alkalinity. So it's quite a range of riverine end members that leads to, we think, quite a range of behaviors. So this led us to think that, to conclude that the Chesapeake Bay is really this nice natural laboratory for studying diverse estuarine carbon and alkalinity dynamics. And we built our proposal on two concepts, and that was one of them. The second is that big things, we think, are visible things, are responsible for sources and sinks of carbon and alkalinity in the Chesapeake Bay and probably in many estuaries. Things like benthic fauna, bivalves, tidal wetlands can exchange carbon and alkalinity with their neighboring estuaries, and submerged aquatic vegetation can play an important role in carbon cycling in estuarine systems. And one example from our 2020 alkalinity paper is given for the Potomac. We constructed a little mass balance model just using the data for the implied calcium carbonate formation, basically, from the alkalinity sink. Because as I noted earlier, you can change the alkalinity by changing the amount of calcium ion. And this blue line here shows a time series of our computed calcium carbonate formation. And it's quite considerable. It's like, it's about 1 kilogram per meter squared per year. That's a lot of calcium carbonate being, presumably laid down every year. And we really puzzled over what that might be. And one hypothesis was that it could be due to bivalves. And this was suggested by one of the PIs on the project, Laura Harris. And we looked into it, and we got some biomass data from the Chesapeake Bay program, and we applied a very simple calcification model to the benthic biomass data, and we came up with these dots, which certainly are quite scattered and only from 2 stations in the region. But it led us to believe that, these fluxes, this calcium carbonate flux could certainly be due to bivalves. And we thought we should investigate that more. Problem is that, bivalves and benthic fauna and macrobiota in general are generally not included in estuarine biogeochemical models. A typical estuarine coastal biogeochemical model will include nutrients, phytoplankton, and zooplankton. I'm not saying that all models only include these things, but many do. And we thought there needed to be more investigation of the role of macrobiota in estuarine carbon cycling. And so that's really the main driver for the CHALK project. And our overarching hypothesis is that spatial and temporal variability of estuarine dissolved inorganic carbon and alkalinity are significantly influenced by these macrobiota. And we're using the Chesapeake Bay as our system to investigate it. And we have a number of sub-hypotheses under this. I won't go through them all, but they involve tidal wetlands, they involve benthic fauna or submerged aquatic vegetation, involve notions of seasonality, and it addresses some of these issues about the alkalinity of the rivers draining into the tributaries, and is also the extent to which the estuaries have fringing tidal marshes. So so that's sort of the, maybe I'll stop there just to kind of give the background and make sure all the audio visuals going well, and if there are any questions that have come up. Janet, yeah.
Yeah, I don't have any questions either in the chat or in the Q&A. And
Okay.
looks like the visuals are going very well.
All right, great. All right, so the Chalk project focuses on these two contrasting tidal tributaries of the Chesapeake Bay that I referred to when showing the alkalinity salinity plot and the related plot there. So so these estuaries differ by the amount of alkalinity and dissolved inorganic carbon that come into them at the head of tide, here for the Potomac, and here for the Mattaponi and Pamunkey, which are two tributaries of the York that merge to form the main stem of the York. Yeah, the alkalinity and dissolved inorganic carbon are much lower in the York, at the head of tide for the York. As I said earlier, there's likely an alkalinity sink there in the Potomac River estuary, and in the York, there's likely an alkalinity source. The York is also distinguished by the fact that it has a very large ratio for estuaries of wetland area to estuarine area. So it's a great system to investigate the impacts of tidal wetlands on the carbonate chemistry. So our project, it involves sampling in these two systems, making CO2 system measurements, which really there aren't many that have been done in either system, particularly the Potomac, sort of high quality CO2 system measurements that people like Janet and others make. We're making measurements of benthic fauna, particularly trying to capture the seasonality, which is not very well known in the estuaries. And then we're doing macrobiota flux measurements for submerged aquatic vegetation, tidal wetlands, and benthic fauna. And then we use those measurements to help develop sort of flux maps that we can use as input to three-dimensional biogeochemical models developed by the folks at VIMS, mainly Margie Friedrich's group. We're also so there's modeling, 3D modeling involved. There's historical data analysis involved, so we continue some of the work that I showed earlier. And then these all have relevance for the hypotheses that we're trying to evaluate. And then we'll also take a look at some other systems, of course, not in much depth, but try to place the Chesapeake results in a larger context by looking at available data from other estuaries that will be part of a meta-analysis. Okay, so I'm now going to, I guess in the next about 20 minutes or so, run through some of the results that we've had so far. And there are a lot, and I probably won't be able to get through all of it, but I'll try to at least give you a flavor for some of the things that we're investigating and some of the things that we're learning. So I'll start with the historical analysis. Most of that work's being done at Penn State. I know it best. And we have somewhat of a history of doing it because going back to our papers in 2020 about it. What we looked at are the seven major tributaries entering the Chesapeake. The carbonate chemistry of that water at the head of tide, at the fall line, coming in, looking at historical data to try to gain some understanding of the controls on it. And so one question we wanted to answer right away was, why does the alkalinity differ so much between the rivers? Why is the York like almost an order of magnitude lower than the Potomac? And then how do other important carbonate system variables vary spatially and seasonally among the rivers, like DIC, the partial pressure of CO2, the pH, the carbonate ion. And then I won't be able to go through all these, but I will show some results for the saturation state of aragonite, which I thought might be of most interest to this group. So this work was done by Riley Westman and Ted Stetz. Riley's at North Carolina State University as an undergraduate. Ted Stetz is a chemist and ecologist at USGS. We also had help from other Penn State researchers like Lee and her grad students in the Department of Civil Environmental Engineering here at Penn State. And so we got water chemistry data from the USGS. It's mostly calcium, alkalinity, pH, and water temperature, as well as discharge. We computed DIC from pH and alkalinity. And then we filled in the gaps using this technique called WRTDS, which is effectively an interpolation routine. We did that for alkalinity, DIC, and calcium. Then we went back and we calculated everything else so we could get a complete time series for everything else. And I'll show you an example here for some of the output using alkalinity. So the point measurements are the dots, and then the sort of interpolated values, smoothed values are the solid line. In this case, you can see a lot of seasonality and alkalinity for the Potomac. I think this is in the Potomac River. Sorry for these units. They're not micromoles per kilogram, but the main point is just to show you how WRTDS, what it sort of puts out. And in this case, you could see an increasing long-term trend in the alkalinity. So Riley learned pretty quickly that the alkalinity, this is the long-term average alkalinity, can be very well predicted from the carbonate content of the soils and the rocks in the watershed. So, and this made a lot of sense to us because carbonate weathering will produce calcium ion and therefore produce alkalinity. So that was a very nice result, fairly clean. Another great result that Riley had was he looked at the calcium data, and sure enough, the alkalinity was very close to twice the calcium ion concentration. So all the alkalinity in the river was really carbonate alkalinity associated with calcium carbonate dissolution. And this is the results for saturation state, for saturation index here for aragonite. You can see that this is a log scale. So you can see there's really tremendous differences among the 7 rivers. There's quite a lot of seasonality here. Most of the rivers are undersaturated, but the Potomac actually is supersaturated, particularly during the summertime. And another thing we found out is that there's a very strong control of discharge on the saturation state. And this was the same thing that we found for alkalinity, that high discharge was associated with low alkalinity, and we're finding it's associated with low aragonite. And so the reason why these differ so much here is not only is the calcium varying along this continuum of rivers, but the carbonate ion also varies quite a bit. In fact, this varies even more so than the calcium ion. So we can really explain why exactly the omega is varying so dramatically among the rivers. Okay, another example of historical data analysis we did was looking at the air-water CO2 exchange in the upper Potomac River estuary. And, it's an important but poorly constrained component in estuarine carbon budgets. And our solution to this was to exploit historical pH and alkalinity data to fill gaps in surface water PCO2 and air-water CO2 exchange. And so we looked at several sites here in the upper Potomac River, computed the PCO2, and computed the air-sea flux using kind of a standard formulation, a wind-driven sort of gas transfer formulation. So we got estimates of the flux that way. This is the PCO2 variation as you go down the estuary here. This is going down the estuary. And here with season, this is the climatology. You can see these are very large values of the partial pressure of carbon dioxide, which is the atmospheric values about 450 or so. So these are very highly supersaturated waters, and particularly during the summertime, the late summer. And the flux per unit area out of this very small area is like 10 or 15 times larger than it is from the main stem bay. This is earlier work we did in the main stem bay, shown here in yellow. So even though this area is quite small, it actually outgasses almost as much carbon dioxide as the whole main stem bay. So these tidal fresh and oligohaline areas can really play an important role in the carbon budget. All right, are there any questions about any of the historical data analysis we did?
I don't see any questions or hands popping up.
Okay, I'll go on to some going from the water quality data to benthic biomass data in the Chesapeake Bay. So we decided to look at the whole main stem bay. The project only required us to look at the York and the Potomac, but since we had the whole data set, we thought we would analyze it because it hadn't really been very systematically analyzed. So we looked at the benthic biomass data from the Chesapeake Bay program. It's mostly summertime sampling. This is a long data set. It's almost 30 years worth of data, more than 8,000 individual ponar grabs from the bottom of the water. And so we wanted to just know some simple things like which species contribute most to the biomass, what's the spatial distribution, and what factors determine the distribution. And so this is work led by Sheyi Ajayi. He's a postdoc in my lab. And I should say that the prior work that I showed was from Maria Herman. She's research faculty in my group here at Penn State. So this is Sheyi's analysis of the benthic samples, and it's mostly bivalves in the bottom. Almost 90% is bivalves, and of those, most of it is Rangia, which is a small bivalve. And there's some of this invasive clam, the Asiatic clam, as well, Corbicula, as well as Macoma, and some other bivalve species. In terms of the spatial distribution, what Sheyi found out is that there are these hot spots of biomass in the Potomac, maybe to some extent in the James, and certainly in the upper main stem bay, all in these sort of upper reaches of the tributaries. And when you separate the biomass by salinity class, going from tidal fresh to polyhaline, you see that most of the biomass is in the tidal, or the highest concentrations of biomass, I should say, are in the tidal fresh and oligohaline waters. We also wanted to understand what's controlling the distribution of biomass. So we developed a simple statistical model using the generalized additive model approach. And it's a very simple equation where you have a response variable as a function of smooth functions of predictor variables. And here's a so-called partial plot for GAMS, where biomass is being predicted from salinity. And it shows the salinity data down here. So you can see quite a large range of salinities are covered. It shows the smooth function. It shows the mean of the response variable here, and it shows the 95% confidence interval. So you can see, for example, here, kind of as we suggested from the earlier analysis, that you get higher biomass at lower salinities here. But we could do that for many variables because Chesapeake Bay is really data rich. So we saw that biomass increased with nitrate, increased slightly with phosphate and then started to decrease at very high levels. There's the salinity plot I showed before. There's also impacts of suspended solids and water clarity. And we're able to explain about 40% of the variability in these data, which is we're pretty happy with. All right, I want to go on to some of the field measurements that we made as part of CHALK. And this is work led by Amber Hardison and her research group at VIMS, particularly her grad student, Novia Mann. So we did some sampling. We're doing seasonal sampling of CO2 system parameters in the Potomac here, and in the York River estuary. So we're out there for 2 years. We have about a year's worth of data now, and this is explaining some of the sample collection. These are slides directly from Novia's recent talk at the Chesapeake community meeting a couple of weeks ago. You can see the measurement protocols used here were dissolved in organic carbon. And here are some of the results shown as a function of distance for the Potomac in orange and the York in blue. And already, you can see quite large differences between the estuary. Here's the upstream point. This is downstream point. You see a decreasing trend in DIC as you go downstream, but an increasing one for the York in contrast to the Potomac. But it's often more useful to plot these. Well, it's also useful to plot things versus salinity, so you can see sources and sinks. So that's one season. Here are all the seasons, which show fairly similar results. Some variability. You can see some of the large differences between the two systems. Large range in the Potomac. In the York, the increasing values in the York going downstream. Looks like there's a DIC sink here in the Potomac that might be associated with the outgassing that we saw from Maria's work in the Potomac. And you can see some seasonality here. So we'll be out there in this. We'll be analyzing the spring 2020. 2024 measurements soon to look to see if that continues. So that's some of the DIC results. We're also measuring organic alkalinity, which is rarely measured, but it's very poorly constrained in contrast to the carbonate and borate contributions to alkalinity. It's not a component of CO2 system calculation programs like CO2 Sys and CCARB, which you may be familiar with. And ignoring an organic alkalinity can introduce errors in CO2 system calculation. So we wanted to measure organic alkalinity and evaluate its impact on the carbonate system, as well as this. I won't be showing this, but to look at, see if we can evaluate different CO2 dissociation constants at low salinities, which have a lot of uncertainty now. So this is work led by Alec Wang at Woods Hole and his lab. Sophie Kuehl is a grad student in his lab. She's done a lot of the work. This is the system that they use to measure organic alkalinity. They basically measure the carbonate and borate alkalinity, and then are able to infer what the organic alkalinity is from that. And this gives some of the accuracy that they have or precision that they have in their lab. And here are some of the results. Organic alkalinity seems to vary with salinity, tends to decrease as the salinity increases. It tends to be higher in the York than in the Potomac. And, it contributes about 2%, up to 2% of the total alkalinity in the Potomac, and as much as almost 20% of the total alkalinity in the York. And then Alec and Sophie did some calculations of PCO2 and pH with and without the organic alkalinity contribution, and they computed it from DIC and total alkalinity. So these are basically the errors you would get if you ignored organic alkalinity. And it can be up to several tenths of a pH unit and several hundred to even 1,000 microatmospheres of PCO2. So quite important to consider when trying to understand the carbonate system. All right, I'll want to talk a little bit. Last set of measurements here are the measurements of fluxes from macrobiota to the water column. We've been doing Emily Revest's lab at VIMS, and her student Javi Pujols have been doing benthic fauna incubations. Cassie Gerbich from St. Mary's College of Maryland has been doing some in situ measurements of fluxes for submerged aquatic vegetation. I'm not going to, for lack of time, describe some of these, but I will show you some of the tidal Marsh Creek lateral fluxes that Jill Areola here, she's a research faculty at Penn State, has been doing with help from a lot of folks from really many of the partner institutions, but particularly VIMS, which is right nearby our sampling site, which is here in the York River. It's Tascanis Creek here. This shows sort of the watershed. This is marsh here. You can see the tidal creek there. It's surrounded by lovely forest, really a beautiful spot. This shows a digital elevation model of the marsh. So it's kind of mostly in pink, and then you can see the surrounding topography there. We did our sampling here, kind of zooming in. We have a current meter located right here, and we do a sampling from a dock there. There's the current meter. Submerged, it's an acoustic Doppler current profiler. And then we have 24-hour, sometimes 12 hours if it's too cold, sampling at a dock here as part of the National Estuarine Research Reserve System. This goes through some of the details of the field sampling, which I'm running out of time, so I can't go through it all. But yeah, we're sampling at the dock there. We're collecting samples for DOC, DIC, calcium, POC, and alkalinity, bringing them back to the lab. And here are some of the results. This is showing the salinity in the dashed line here. So this is like low tide here, low salinity as the tide's coming out. And you can see the alkalinity is really high. And on the incoming tide, the alkalinity is quite low. This is very large, several hundred micromolar difference in alkalinity. This is very clearly telling us, at least for this season here, that the marsh is exporting alkalinity. And it turns out, so this is the summer shown again in green, and then we have our fall measurements here, and our winter measurements here are only out for 12 hours. And you can see while the baseline alkalinity differs, the range really doesn't over the tidal cycle. And so we're pretty confident that the salt marsh is a source of alkalinity to the neighboring estuary, and it's probably happening via sulfate reduction in marsh soils. Last bit, if I could just take a couple of more minutes and just show some of the modeling that's been done by folks at VIMS, led by Margie Friedrichs. It's students in her group. First, as a student who graduated recently, Fei Da, he did some detailed high-resolution studies of the York River, three-dimensional biogeochemical model, forced, at its boundaries. And this shows some of the model evaluation. They pay a lot of attention to model skill. And he did a series of experiments showing impacts of hydrology, tides, and wetlands. And I think I'll just show this result for lack of time, running out of time. This shows a DIC budget for the estuary. And the main thing to point out is that of the sort of biological inputs or lateral inputs or internal inputs, tidal wetlands contribute quite a lot. Say compared to the water column biology here compared to the lateral fluxes from tidal wetlands. Tidal wetlands either match the biological inputs, which, this system is net heterotrophic, so there's a net input of DIC from biological processes, and wetlands export DIC almost as much, or sometimes quite a bit more. So he also looked at impacts of tides and hydrology, but I don't have time to show that. So I'm going to go on to Alexa Labossiere's work, and she looked at the whole bay here and focused on the impacts of tropical cyclones. And here's an example study where she looked at the impact of Hurricane Isabel on alkalinity in the Chesapeake Bay. And so here's the river discharge associated with the hurricane. Here's the peak in winds associated with the hurricane, and she teased apart the individual effects of those two. It turns out the river discharge generally plays a larger part. And, you know, she ran experiments with and without these peaks in river discharge and winds. So there's kind of hurricane and no hurricane experiments to really separate the impacts. And the impacts are quite dramatic here. This is the alkalinity at a site as a function of time, and you can see how much the alkalinity drops in response to, in both the York and the Potomac, in response to this hurricane, bringing in low alkalinity water from land. This shows a movie of those results. And really big impacts on the outgassing. So this is showing the air-sea CO2 flux with and without the outgassing. Negative means outgassing. And so you can see, you know, when you have a hurricane, you have actually quite a lot more outgassing than when you don't have a hurricane. And it's sort of a movie of that as well. The bottom line is this is the fraction of the yearly flux of CO2 just contributed by the hurricane. So I'll just put your attention to the black bars here. When you have the hurricane, you're getting, you know, depending on the river, 25 to 35% of the annual flux just from that several week period associated with the hurricane and its aftermath. So I'll end there. I'll just go back to our original slide that kind of summarizes the project. I apologize that I don't have a picture of everybody on the team. I just didn't have time to include that. That'll be in future presentations, but it's really been a great group to work with. I'm really privileged to be working with everybody with such diverse expertise in measurements, modeling, and data analysis. So thanks very much. I'm happy to take questions over the next 10 minutes.
Thank you very much, Ray. It's really great to see all of this new data coming out of the tributaries of the Chesapeake. And I know that you guys are just kind of starting to scratch the surface with your results. So I do have one question so far. Can you comment on the carbonate alkalinity measurements in the rivers and organic alkalinity? Do those measurements take into account the organic alkalinity that you mentioned farther downstream? And if not, how does that impact your overall budget?
So I think, okay, so for the historical data, we don't have estimates of organic alkalinity. So we have errors in our computed quantities that I showed earlier because we ignore the organic alkalinity. And those errors will be on the order of what Alec showed in his analysis. So our calculations so far, like in the model, do not include organic alkalinity. That's something that we want to be able to include. And that's why the measurements from Alec's lab are so important. So the plots that, for example, I just showed for Alexa will change to some extent when we include a parameterization for organic alkalinity. And, you know, I think they can be substantial. We did for the whole main stem bay, we did an estimate of the impact on the outgassing. You know, it wasn't dramatic for the whole main stem as a whole, but it could be significant in certain places at certain times.
Thank you. So the next question we have is first, thank you. And then I wonder if you and collaborators are planning to measure any particulate inorganic carbon from rivers and SAV. These sources of alkalinity could play an important role in carbon and alkalinity cycles across the land-ocean interface.
Yeah. No, that's you're identifying you know, a limitation of our study, and we're not doing those measurements. And I know PIC can be important. There was a recent study in the I think the ALEC was involved with for the Mississippi, where the PIC dynamics seem to play a role. I will say that Cassie, to her, you know, she had the insight to at least collect the calcifiers, and any calcification, at least that was visible. I think she's just down to the organisms. So she's scraping those guys off of the SAV, collecting them and weighing them. Although I don't think she's actually measuring the carbon content. So that's the degree to which we're doing it. And if the questioner wants to join us on our, you know, we have another year's work of field work, and if they're doing any PIC measurements and would like to join our campaign, we'd love to have them.
Well, I'll just throw it out there that it was Faye Daw that asked the question.
Oh, okay.
So
Yeah.
sorry if I just put you completely on the spot, but
That's okay. You know, I'm sure he's okay with it. Yeah. Maybe he knows someone that will do it, or yeah.
Well, I guess contact Ray directly, but MACAN is always here to be a conduit for collaboration as well. I don't have any other questions that have been asked, and I don't see any other hands up right now. So I'll just give folks another minute while I also just let you all know that in a few short weeks, MACAN is going to be putting out some educational videos that some of you may have even participated in over the last year. They'll be on our website, and we'll be sending out an email to announce their arrival. So please watch your email for that, and also watch your email. We do plan on having one more webinar this year. We're not exactly sure when it's going to be. It's going to be our MACAN fellows that will be presenting. So watch your email for that. And I still don't have any other questions or hands. So thank you very much, Ray, for joining us today. This is really exciting. And I know that you provided some links to PDFs if anyone wants those. They're also available on the MACAN website, because we go around and download PDFs and put them up there. So you can find them there, or ask Ray. I'm sure he'll be happy to provide them, or ask us. We'll get in touch with Ray and ask them to be provided. So thank you all again. Oh, wait, I do have one. Oh, thank you, Libby, for the exciting comments and exciting work. And Robert.
Thank you, Rob. Yeah.
Thank you all very much for joining us today. And this webinar will be posted on our website shortly. So, thank you. Have a great rest of your day.